Hello, everybody. Welcome to the iPredalis podcast. We're a member of the Chameleon Academy Network, and today's episode is a conversation with Dr. Ben Morrill. Bill Strand had us on to his podcast probably back in 2021 to introduce this project of testing Panther Chameleon locales based on a 2015 study using baseline data from animals collected in the wild that never entered the pet trade, and then using the markers from that study to track the lineage of the animals that we now have in captivity. If you haven't listened to that episode, I highly encourage you to check that out because Bill stops us and asks many great questions. I think that's a really good conversation to start with. This 30-minute segment is an announcement of the final round of testing, and in many ways assumes that you've heard that first episode. So I encourage you to check that one out first if you haven't, and then join us here in a update about what we're going to do in the next few months as we do a final round of testing and release a simple version of the test for everyone to start testing their animals and get some feedback on what their genetics say about their local origin. Once Ben has collected a number of sheds from many different local forums, his goal is to do a more comprehensive full genome test where he's sequencing the entire genome of all the Panther Chameleon submissions that he gets, and then he takes the markers from the 2015 study and finds microsatellites that are correlated with the known markers from the wild specimens in the 2015 study based on the samples that are coming in and then the analytics that they are going to be doing on the full genome sequences of the tests that we get. And Dr. Ben Morrill says it better than me, so I hope that you enjoy this conversation. And over the coming months, as the new submissions come in, I'd be happy to hear what your thoughts are, what your questions are. And I know that there's going to be some ideas that come out of the community that guide Dr. Morrill as to which direction the market is going to have the most demand for his services. So I highly encourage you to reach out to him, reach out to me, and to provide feedback on some of the ideas that you have for what we should do. So I hope you enjoy this conversation, and I'll catch you later. This is iPredalis. So welcome, Dr. Ben Morrill. It's really good to have you.
Thank you. Glad to be here.
Yeah, I think we made an exciting announcement about 2 weeks ago now, and I wanted to just have you on to talk a little bit more about the opportunity and what's coming. So
Yeah. So we've continued like we've talked before. I think on here, definitely to each other about the ideal test we want to have, which will take still some more work. Every time we make progress, then we have another hill to climb that pops up. But we definitely still can use more sheds when we can do the full. We're going to call it like a genomic registry test. So right now, the few sheds that did come in, the testing I did was just the few spots where they had in that publication where they looked at all the panther chameleons through the whole distribution of their natural range. So we can definitely use more sheds, and we can still do that simple test. And so, like I said, to you, another 20 to 30 would be totally fine to have people send in, and there's still not going to be any additional charge. We'll run those, get people results. Most of the time, it'll be within a month of when the shed comes in, unless we just did a run or something weird like that, and maybe it'd be a little
Like
longer than
the
that.
time you just get bad timing,
Yeah,
but
yeah.
it's like,
Most of the time, it'll be a month or less.
mm-hmm,
But what we'll do is we'll use the sheds. I still have the sheds and some of the DNA from the original 20 or so that came in. I'll keep
mm-hmm.
that from these as well. And then when we can do that genomic registry, that gives me an even better, broader base to be able to make that test from. And so the only thing we ask, I think we were talking about limiting it for each person, to only a couple or something like that. But what
Yeah,
we don't want like 10 animals that all came from the same pairing, because that won't help us design a better test. We want like as much
exactly.
variety as we can.
Yeah, we want to stratify it across the locales and across pairings and everything. Yeah. So that's part of why I was just like, will just start with getting a submission of all the people who have stuff that is ready to go, and then on your side, you could basically, we're gonna take the results from what's been submitted, select the people who we want to invite to submit the free ones, and we'll be able to then sift through it, make sure we get a good sample. Um, but there's no harm in submitting, just because you never know if you're like the only person with Tamatov, and we don't have any Tamatov yet. Even if there's like 50 submissions, we're still going to want the Tamatov. So I feel like that's part of it, is just, if you've got something, submit it, let us know, so then we can do a stratified sample of it. Is that a decent way to think about it?
Yes, yeah. And as far as when you're collecting sheds, I know some people still have questions about, how fresh it needs to be and how do you collect it and when would it potentially be bad or whatever.
Yeah,
You just have to remember the number one thing in your mind is that
keep it
moisture,
dry.
humidity, wetness, that's what destroys the DNA. And so
Mm-hmm.
just if you know the animal is close to shedding, just check every morning and night, try to get uh, and really, a piece like the size of a nickel to a
A quarter.
quarter is really all that we need. And make sure once you collect it that you let it get completely dry before you put it in a Ziploc. Once it's
Mm-hmm.
completely 100% dry and it goes in a Ziploc bag, you can write the animal information on it.
Yeah.
But once it's in that
Yep.
bag, even at room temperature, I've run them after 6 or 7 years and they still work just fine. So don't
Perfect.
think, oh, I gotta, wait till I know that it's
Yeah.
my shed is a good,
Yeah.
one that they want to run. Like, no, just go ahead and collect it. Collect it,
Yeah.
get it completely dry, put it in a Ziploc, and whether you send it in today or in a year, yeah, just like that. Just I know some people they have like a little file cabinet and they have just a folder there that's just a shed from each of their breeders and they just have it there. If a test pops up they want to be able to run it for, they can just take a little piece off and send it in.
Send
However
it
you
in.
want to
Yeah.
do it. But the
Yeah. And
sheds
with
stay good for a long time.
cool. Yeah. And for chameleons, we're lucky because they're dry shedders. So
Good.
they come off dry. If you can get them before they get misted, then um, we don't have too much worry about the moisture level on it. It's not like they're going to
Yes,
soak and then shed after they have been soaking for a while. So
that is lucky. And
yeah.
the other thing, especially with lizards, people ask, well, they always shed in pieces. What if I don't have a piece the size of a nickel or a quarter? As long as you
Mm-hmm.
have pieces enough to cover a nickel or a quarter, it's okay if they're torn. The first thing we'll do when we get your shed in is we'll tear little pieces off. That's part of the process. We have to break it down. So it's totally fine if it's 30 pieces that are all really small, but it's enough to cover, the top of a
Yeah.
nickel or a quarter. That's great.
Yeah. Cumulatively covers it. Yeah.
Yeah.
Cool. Okay. And then once we have, so that's probably going to be about a month before all those come in, because it takes time for them to shed, takes time for those samples to come in. What should we expect next?
So yeah, once the sheds are here, like I said, most of the time it'll be about a month, and then we can have
Oh,
results for you. And then once we've run that additional, 20 to 30, and then can stack that on top of the ones we already have. We've started very first with the ball python and put a lot of money and time into that. I'm hoping over the next 2 months that we'll have the genomic registry working for ball python. So if you see on Morph Market or in social media or on our YouTube channel or whatever, hey, we've got it for
Yeah.
ball pythons, that means that all that time and money that went into getting it to work in ball pythons, we'll be able to turn around and use it for other species much faster than this. This first one's the biggest pain in the butt because we've got to work out all the kinks. But as
Yeah.
soon as we know how to make it all work and be fast enough and cheap enough that it'll be helpful. If we don't mind waiting a year or two, then I can do it pretty cheap, but that's not really a good way to run a business. We can't wait that long, but we also can't charge $500. So yeah, we had to figure out a way to kind of balance all of that, being fast enough, but also cheap enough. And I'm pretty sure over the next couple of months, but I've thought this like at least 5 times, and then something else pops up that we have to solve. So
Yeah.
don't quote
No,
me on it, but
I know I've mentioned that Instagram post that you did running on the beach. Do you remember that one where
yeah.
you're running on the beach like sand in your beard and trying to keep up with the volume of tests coming in? I thought that
Yes.
was hilarious. I thought that was hilarious, but I used that as an anecdote to be like, he's working hard, guys. But
Yes.
it's just it's complicated, and there's a lot of things that you got to work through to make it accurate. You don't want to start
Yeah.
just sending stuff out that people can't trust. So
Yeah. Yeah.
that it's like and you won't know until you got results. So there's a
Yeah.
lot of kind of steps that you got to go through, and it's the first step is collect sheds. We need more sheds.
Yes.
So I
Never
think
go
that's
wrong with that. Save them up.
yeah. Because the more data we get, the more runs we do, the better we get, and
Yeah.
it'll continue to improve over time. So that was the next thing I kind of wanted to get into is like the first results that we're going to get are just checking for microsatellites that were published in that paper, right? It's just like we
Yeah.
know that these are found in this region and nowhere else. Therefore, if the shed that you send in has them, there's a good chance it's from that region. Um, whereas the way that we're gonna, do you want to talk a little bit about that by itself, and then possibly like what the full genomic test will do to improve upon that?
Yeah. So in the part of biology where we study this kind of stuff, taxonomy or phylogenetics, um,
Mm-hmm.
it's changed a lot. Like 50, 60 years ago, we're only looking at, ones from this area are green, ones from this area are yellow. This has more scales than this one. It's all stuff on the outside. And then
Mm-hmm.
DNA came, and so we were able to start looking at, okay, here's some DNA, and we look at the DNA from here versus here, we can tell a difference. And then it was, okay, we want some nuclear DNA and some mitochondrial DNA, not just mitochondrial. And then,
Mm-hmm.
it's gotten to where, we want more and more of the genetic information to be able to compare whether it's different locations or different species or subspecies. So today, the test that we'll do with these sheds right away is
Mm-hmm.
fairly modern. We're going to look at multiple genes, and some are mitochondrial, some are nuclear. And the details of that, feel free to look it up, or we can talk about it another time. But
Yeah,
so
yeah.
it's pretty good. And in that publication, they were able to show some distinct differences between the localities, which honestly surprised me just seeing panthers. I wouldn't have expected they could find. Didn't it end up being like 10 or 11 different groups or something like that?
Yep.
I mean, that's
Yep.
pretty impressive. And then they have these trees in that publication where it shows,
The
the length of
distance.
each branch. That's how
Yep.
many different changes there are that only exist in that group. And some of them, it's like 7 or 11 differences, and it's
Yeah. The
pretty
biggest
impressive.
difference I think was 21.
Yeah, I mean that's
And so I
crazy.
was like, yeah, it's a lot. I was like, wow, okay, because each one of those takes a really long time for the populations to be separate for that to
Yes.
for that many to occur, right? So um,
So yeah,
that that I
with that test, we're looking at like 2,000-3,000 base pairs. With this genomic registry, what we'll do is we will take at least these first 50 that we have. We might need more. I won't know till we look at those first 50. But we're going to sequence all 50 of those. Even if we've already given you the information for the smaller test, we'll rerun it once we can do this whole genomic registry. And then we're looking at the whole genome. So we're looking at billions of base pairs. And what we'll do is we have someone working with us. We're very lucky. This person has been a huge help doing the bioinformatics. So the data
nice.
side of it. Yeah, where you're looking at billions and billions of base pairs.
You got a Python programmer?
Yeah. He's extremely good.
Very
Yes.
cool.
He
I love
happens
coding
to
too.
breed. He happens to breed reptiles, so we got lucky, and he's interested in helping us out. And he's a very busy person with a very high caliber job, but he's willing to help out. And a few people got to meet him at Tinley, and maybe as we do more and more of these things, I can get him to come to more shows. He's awesome. But what he'll do is he'll take all those 50. So like with ball pythons, what he's done, we've taken like probably close to 1,000 now. Different individuals, and we have 5 million different variable markers throughout the whole 1.5 billion base pairs. And so that's where we're at with ball python. So it's like extremely easy for us to tell you, if you give me a piece of shed skin and say, hey, this guy said he tested this and it's this and that, is this the same animal? And I can tell you times 10 to the minus 500 whether it's the same animal or not.
Yeah,
So
flippers beware. Yeah,
it's very powerful. It's
I
also,
like
and
that.
some people brought up the fact, like, if you have a high end collection and this is like something you're doing as a business, even if you had animals stolen, not only could we identify your animal, but any offspring from that animal, we would have very high knowledge. Hey, this is an offspring from that. It's the exact same technology they used to find the Golden State Killer.
Yeah, like forensics.
Yeah.
It's a
yes. Yeah, it's very powerful. So right now, today, we don't have that yet because we haven't run this first 50. So we've got to get the sheds in. We'll do the simple test first. We'll run all 50, and we'll have a whole genome. We'll have that registered. It'll be part of Rare Genetics Inc. over time. It's kind of like 23andMe, things like that, where every few months you'll get an email. If any of you have done that, you'll get an email and say, hey, we just updated this. This is the updated information about you. So like you all that get this at, free, this first 50 or whatever, we'll always have that information. So 10 years from now, you might get an email from us and say, hey, now that we've run, 7,000 Panther chameleons, we can tell you this and this and this.
You thought your ambilobe was pure, but it's actually not. Like, oh, thanks. 10 years later.
Well, or we could say, hey, you had these 3 females registered. This one right here, it has some traits that are some genetics that show that it has a higher. It will have a higher egg laying, or it's more likely to start laying eggs at a younger age, or it has genes that mean it's going to have a longer life. There's a lot of different things.
There's a lot of cool stuff. Yeah.
Yeah. And tons.
And one of the things I was thinking about that is like as you improve upon it, we just continue to learn more. And if people have the right mindset, then they're like, oh, cool. How do you collect some of the, I guess, corollary data related to, say, clutch sizes or the phenotype, the way that it appears compared to the genotype that you're getting information on? Are you going to ask for more data from people? Say they're part of this first 50 and they send in a female shed. You want to know details if they're going to share, say, like clutch sizes and things like that?
Yeah, so right off the bat, we probably won't for the first 6 months or whatever. But yeah, it could be that a lot of the times it's breeder driven. So like somebody will say, hey, my females always lay at the first 3 months of the summer. And then someone else says, well, mine always lay the last 3 months. And they're like, okay, you live
Let's take a
here,
look
I
at
live
that.
there. Like, is it, you could first of all see, okay, if all the breeders in California go earlier than the breeders in, Oklahoma, maybe it's where you live. Then if you say,
Mm-hmm.
okay, but me and my buddy live in the same place, mine go earlier or later. Okay, well, let's look at the genetics. Or it could be clutch size, or it could be longevity, or, any of those
Yep.
questions. And then what we
Yep.
do, if we have a couple of people that say, hey, mine are like this, mine are like this, then we could do things like have them, trade some animals, have them in the same environment. So this all
Yeah.
takes time. None of this is like, hey, next week we're gonna have an
No.
answer for you.
Oh yeah.
But
No. And for Panther Chameleons, we're going to be more in like the leopard gecko world, um, where it's a lot more polygenic traits, a lot,
mhm.
and not as many dominant,
Yeah,
co-dominant
like
type of
single gene
trait
things.
like
Yeah,
single gene things. And so whenever anybody has asked me about the ability to detect color, I'm always like, that one is hard because of the
yeah,
fact that the genotype is complex. Like it would be an amazing discovery if we can nail down, the whatever number of genes it is that encodes for a given phenotype, but
yep,
we know that it's very complex,
oh
um,
yeah.
and so that's at least my base expectation is that part will. I mean, maybe you guys have some experience with some polygenic traits where you've nailed it, and then we can start to learn from that. But
Yeah, I
um,
mean, for an example to kind of bolster what you're saying in the human genome, where there's been billions and billions of dollars spent, just in the last year, they came out with a publication where they did a genetic analysis, so polygenic trait of height, so how
yeah,
tall you're going to be as a human, and they were able to isolate 10,000 different genes that play a role in
how tall you're going to be?
yeah, it'll essentially affect how tall you're going to be. So for color, like if you just focused on blue, maybe there's only 3 genes that really play a big role, or there could be 30, or there could be 300, and we don't
Mm-hmm.
know till we start looking. But if that's
Yeah,
something that you as Panther chameleon breeders are really interested in, and somebody focuses and says, hey, you seem to have a lot of red, or you seem to have a lot of yellow, and it doesn't seem to follow, especially if it doesn't seem to follow locality. Like if it always follows locality, that kind of makes it a little easier. But if all of a
yeah,
sudden one pops up that by color really should be a different locality, but from all the genetics we've done it doesn't match, then that's a really valuable animal, because then we can look at that and say, okay, this one's got a color
what
it
happened?
shouldn't.
Yeah, yeah, like what
Why?
happened there? Yeah.
Exactly. And so over time, as we have more and more genetic information coming in, we don't even have to like get more sheds from people. We might 3 years from now, we might say, okay, we think we have 3 candidate genes that seem to give us more blue, or increase your probability of having more than 50% of the animal be blue, or however we draw those lines. And then we might reach out and say, okay, all the people that sent in their sheds over the last 3 years, we'll shoot them an email and say, hey, take a picture or tell us the percent of the body coverage, and then we'll be able to fill in that data for those 500 animals or whatever. And then we can say, okay,
Yeah.
those 3 things, do they really explain? And then we'll be able to test it, once we have that information back from people. So there's a lot of ways that you as breeders can kind of help us guide, what we're interested in, what's important. And whether you're breeding
Mm-hmm.
cattle or chickens, or all these different things, a lot of people thankfully have gone through this in food species, and so we have a pretty good framework. When we know what question we want to ask, then we can figure out, once we have a
How do you
bunch of
tackle
genetic
it?
information, yeah, exactly, how to figure out what the answer is. And then if there really is less than 10 that play a role, we can develop a test just for those 10 and say, $50, send it in, we'll let you know, hey, if you're going to breed this female versus this one, which one's more likely to give you a lot more color in the babies, or something like that. That would be
Mm-hmm.
possible.
Yeah, yeah. Panther chameleons are fun. They from so genetically speaking, but also phenotypically speaking, because of the fact that they change color.
Yeah.
Um, they have seasonal. things where they go through seasonal shifts. Um, they sleeping colors are different than when they're awake. They have mating. They have mating colors that they fire for females. Um, so you run down the list of all the different things you could potentially try to learn about them.
Oh yeah.
Uh, genetically, there's tons of stuff there. Also, very very hard to to generalize,
Yeah.
because you make, and I do this all the time. I'll make a statement, a general statement about something, and then someone will say, "But my chameleon's just making you look like an ass. Look at this," and I'll say thanks, but that's just how the species is. It's kind of that's what makes
Yeah.
it fun in a lot of ways. Is that it it's phenotypically and genotypically complex. So I think that that's what's going to make it an exciting project to continue to improve on and continue to add to. It's the kind of thing that I feel like you could spend your entire life on and not finish.
Oh yeah.
so so that's definitely something that I'm excited about related to this project. So um, I guess to close it out, we probably have about 5 more minutes. Um, once what are some of the things that um, you've learned in rolling out tests for other species that you can share with us, just so that people have a good, besides the timeline we just talked about, but maybe some of the other stuff, the right kind of, maybe the best attitude you've seen from some of the people that are participating, what makes your job easier, and then also like how do we do a good job talking to other people in herpetoculture about the work that we're doing. Um, do you have some advice for us on that?
Yeah, I mean, certainly a huge part of it is, how much interest and excitement there is for what we're doing. And so if there's something that's interesting to you and exciting to you, definitely talk about it and have discussions and figure out what's important. We might find out that, what I think, what John thinks, maybe that's not what the majority of people want. Maybe there's something different they want instead or on top of or whatever. Like, for me, I
Yeah.
don't keep chameleons at all. So I don't
Mm-hmm.
have, with ball pythons, I had at least bred ball pythons, so I kind of had an idea. But this I'm a very open mind. However people want to go. Once we have that framework and have sheds coming in, we can go different directions with it. Um, but yeah, so the other really big thing that will help make the testing good fast is having detailed information about each shed and have that be correct. So whenever you're working with sheds, only have one bag open, only have one shed out at a time. Like don't like go through and pull like 7 sheds and have them all drying and then like go and bag. And because what we found is about, on average, probably 2 out of 100, there will be some kind of shed mix up, whether it's on the customer side or our side. So
The
it's not a problem with
contamination,
the
yeah.
test at all. It's, well, a lot of the time, like they sent me a shed in that was clearly a pied ball python that they expected to possibly be het clown. But when I tested it, it was negative for pied, and it was a clown, which they knew it wasn't by the way it looked. And I was like, well, do you have a clown? I mean, they just put the wrong shed in. So, and it could be
Yeah.
as easy as they were in together to breed and then they pulled the one out, but
Pulled
it
the
had
shed.
shed while it
Yeah.
was in there. And that's the piece they pulled. So just be
Yep.
really careful. Like, think about it when you're getting that piece of shed. If two animals have been in the same cage for a while, you want to like actually take that shed off of the animal, or make sure there's no pieces of shed already in the cage. Make sure that's all gone, and so that when that shed, when you see something there, 100% sure that came from that animal. Those are some
Yep.
things, because when we're developing the test, if 5 to 10% of the sheds are messed up, then that makes
Those are the
the
one- those
test.
are the, those are the ones you waste a lot of your time on.
Exactly. The test development will take longer. We'll still get there, but it'll take us longer, because if 5 to 10% is sending us away, like, you do some software stuff, we're essentially taking two groups saying this is, this locality, this is this locality, and let's see what markers we can find that differentiate. Well, if some of them are wrong, then you're throwing some out that are really good, or you're bringing some in that actually don't differentiate the two groups. So that can
Yeah.
slow things down. So yeah, just being really careful. Make sure you get the right shed and make sure you get it labeled correctly. And whether you keep it there for in your filing cabinet or send it to us and we'll keep it in our filing cabinet. Either way, just make sure that the data is good.
Okay. So we'll do our best on that. My guess, my baseline expectation is that in the captive population, the local form is maybe like a coin flip as far as being right. Um, especially based on that 2015 paper, because they tested a bunch of animals in Europe and they came up with less than 50% being pure, or matching what the breeder said it was. Um,
Hmm.
and and so my baseline expectation is that we're going to have a lot of stuff that is mislabeled. Um, so that's, even if you grab the right shed, is
Yeah.
probably going to make that part fun.
Yeah.
I,
Yeah.
I know that I have some wild caught animals that were imported. They have the right phenotype. They look classic, on Bilobe, and I'm going to be sending those in. I know Ryan Robertson has some also phenotypic wild caughts that look exactly how they should. So those things we'll try to make sure we mark those clearly for you,
Yeah.
because those are different. Like if you have an F1 versus a long term CB animal, right?
Yeah. Oh yeah.
Do you'd want to know those types of things? Like this thing is 10 generations captive bred versus something that's pretty fresh.
Yeah. Yeah, that'll be good because the first thing we're going to do for these, first 2 or 3 or maybe even 4 or 5 months, we're just going to compare it to the published data. And so that'll be good to know, if this is several generations, we don't know where it came from and it comes out as being from like 7 different localities and we're not as stressed about it. But if we have
Yeah,
one
yeah,
with really good documentation, it says it's this thing, but it says it's 7 different localities, then we'll be a lot more worried about that test if something got messed up or if there's something really
yeah.
weird going on or whatever. So
Well,
yeah, that information.
then we start reaching out to the importer, and we're, hey, what happened, man? You
Yeah,
know,
I know
that's
with green
seven
tree.
seven.
Yeah, with green tree pythons, I know that there is a researcher that was there, in
Mm-hmm.
Papua New Guinea and watching and Australia some too. But I mean, he pretty much said that he saw stuff come from here, there, and everywhere, and it just wherever it get exported, that's what it said. Oh, it's from this locality, but a lot of the
Yeah,
time people had broken rules to get there's people that have very very little money, and that's a way for them to make money. And
yeah.
if they get
Same
more
deal.
money, yeah,
Yeah, same deal with Panthers, and we don't even have we're even in a worse boat because of the fact that they're sexually dimorphic, and females from anywhere can be thrown in. You have
yeah,
no clue. So yeah, there's that process is messy, and that's part of why we want the test. Also, what makes it hard. Um,
yeah,
I think that, um, the we're blessed with in situ wild specimens,
yes,
as the baseline, but everything after that is kind of, is where the complexity of the test development and everything else, we're gonna have to work through it,
yeah,
explain it, probably just tell a good story when you're finished. Some of the challenges that you've worked through and some of the things that you saw, maybe things that you're like, I'm still not quite sure, we're going to continue to work on, um, that kind of stuff, because I think that will help people, trust it, knowing that we're, honestly wrestling with these very real challenges. And I think that's like, it's the best you can do sometimes, is just be like, hey, we've got really good baseline data and we're working off of that. We're going to
yep,
continue to improve, and the first step is to get these 50 in here and get going. So
yes. Yeah, yeah. Well, I mean, I'll be more than happy. And whether it's coming on here, if you want to come on, our show, or if it's, talking together at Tinley or, one of the shows where a lot of Panther Chameleon breeders are going to be or whatever, I'm more than happy. I'm very much an open book, and people that, have been interested in other testing we're doing, when a test is failing, we've refunded people money and just say, sorry, it's not working. And then some of them 3 months later, we get it working again and we, rerun people's things. Others, like the green tree python sex determination test, we've learned from researchers that there's something funky with that genus. Out of all the
Mm-hmm.
python genera, Morellia has something weird with their sex chromosomes, and I can't do that test. I haven't been able to, and it might not even be possible. So anyway, I definitely will let you know exactly what's going on, and anyone's, welcome. We may not get back to you, especially in the summertime where there's tons of babies and lots of testing going on. We might not get back to you the same day or the next day, but we do. There's 3 or 4 of us now that are answering questions.
Cool.
So yeah, we're happy to discuss, and that's my personal selfish reason for doing this. I think there's a huge amount. I just saw what this kind of stuff did for food animals because I was in an animal science department. I saw what it did for food animals, and every time they're talking about all these cool things they did for chickens and turkey and cattle and all this, I was always thinking about, oh, that would be so cool to do that with monitor lizards or with, this or that. Or
Mm-hmm.
so
Yeah.
I'm excited. We're finally starting to get some things rolling. So
Cool. Well, it's exciting stuff. Um, is there a date where we'll close it off and tell people to stop submitting, or what do you think?
I think once we hit 30, that's when we would say, okay, we've got a good amount.
Okay.
And like I said, if it's not going to be helpful if they're from the same pairing. So, definitely if 2 or 3 people bought animals from the same pairing and be like, oh, I know this one's from that same one. Like, that's not going to help make the test good fast as much as having more diversity. So yeah, like you said earlier, it might be where the first 30 people say, hey, we have these. We might be like, okay, but those 4 are all so closely related. Really just pick one of those 4 and let's find something, that may happen. But I wouldn't say there's really a date, because as soon as the sheds get to me, I'm not going to wait for all 30 to come in. As soon as the sheds get to me, I'm going to run them because I already know how to run that first more simple test. So yeah, as soon as they get here, I'm going to put them on the next run, which is usually within a month, and get that information to people. And then once we have run 30, we have all that information, and we have the ball python genomic registry going. Then we'll run all of those 50 again as a whole genome test, and have the bioinformatics person find all those variable markers for us.
Okay. Cool. So people need to get their name on that list and submit, because if they submit without putting their name on there, then what's the charge? What's the what are we think? Do we have any knowledge about that, or is that still a future?
No, definitely no
detail?
charging yet. Yeah, I don't feel like I know enough yet to feel comfortable
to charge money for it. Yeah,
no charging yet. I would say that, like, once the whole genomic registry is ready, I would say it would be very difficult to make it under $100. But sometimes they'll make really good progress in sequencing or analysis or something that could make it a lot easier for us. But yeah, I would say, around $100, give or take $20, would be a pretty realistic expectation. If we can make it available like in June or something like that, that's probably what that would cost. But we'll see.
Yeah, I think that it's pretty telling how much inflation there has been since the last time we spoke, because it's in the test now, but I get it. We all know how know how it works, and
well, and there's probably 3 or 4 new problems that I know that we have to solve now that I didn't even know about the last time we talked.
that we got to fund. So that's cool.
Yeah.
I think that's totally reasonable, and I know I would be happy to pay that for to test my females. There's no question about it. So,
Cool.
Thank you so much, Benson. Do you have anything more to say to us?
No, I'm looking forward to it. I've kept lots of species of reptiles, and it just happens to be that ball pythons. There's so many people that have them. I know it seems like we're a ball python centered business, but I'm very excited to start doing a whole bunch of other species. And I think 2024 will be the year. I think we'll have 3 or 4, maybe 5 or 6 other species we'll have tests for. So I'm glad you all
Cool.
are excited and interested in being a part of that.
Cool. Thanks, man. So um, we'll be in touch. We'll have to do this again. So
Sounds good.